Welcome to Unit 3. Unit 3 involves Chapters 6 and 7. Chapter 6 deals with the organelles of the cell and their functions, and Chapter 7 deals with the cell membrane, how it's put together, and what it enables cells to be able to do or not do. We already know that cells tend to be very small, although some cells are significantly larger than others. For example, muscle cells can be up to 30 centimeters in length and contain hundreds of nuclei. Regardless, microscopes are used to study them. Cells have been studied for a few hundred years at this point, and it took contributions from several people to develop the cell theory, which is a unifying theme of life. The cell theory has three basic components that applies to all cells as we know it. The three components are, first off, all living things are made up of cells. Secondly, the cell is the simplest collection of matter that can live. And third, cells can only come from pre-existing cells. This chart allows for comparison of the size of a cell to other objects using the metric system. There are 1 million microns, or micrometers, in 1 meter. As we can see in this chart, large cells, around here, are 100 micrometers, which is considerably small when compared to the length of 1 meter, which is about 3 feet in length. Scientists use microscopes to visualize cells too small to see with the naked eye. One of the most basic microscopes are light microscopes, and these are the ones that you have used in school. Light microscopes, or LMs for short, pass light through a specimen, which then goes through a glass lens, which magnifies the object. The quality depends on a few things. First off, quality depends on magnification. The second, resolution, and third, contrast. Magnification is the ratio of an object's image size to its actual size. Basic light microscopes, or LMs, can magnify about 1,000 times. Resolution, that's the measure of the clarity of an object. Microscopes allow us to see the finer details of something that we could not see with the naked eye. For example, a car may appear to have a nice paint job from a distance, but moving closer may prove otherwise. With the light microscope, you have probably been able to point out the nucleus and the cell walls of cells, but beyond that is quite challenging. You'll be able to see a bunch of dots and other items, but being able to do a great job identifying which organelle they are, well, good luck. It's tough. This is in part due to the resolution, but it can be enhanced by adjusting the contrast. Contrasts are the visible differences in the parts of the sample. Different methods of staining cells have been developed that can produce such differences. Prepared slides with different applied contrasts can be purchased for use on an LM to make additional organelles more visible. Going through some of the examples that you'll have for contrast, you can see some unstained cells compared to some stained cells. The nucleus and other features of the cell become a bit more observable once they are stained. A few more examples of staining here and here. Probably one of the cooler ones would be the fluorescence. You'll see this used quite commonly, a bit more artistic, but once again you can see some of the goings-ons. Confocal, once again, another staining image. Alright, moving on. Microscopes I want. These are two electron microscopes, or EMs, that are awesome at seeing the very fine subcellular details of cells. The scanning electron microscopes, or the SEMs, which are the top ones, focus beams of electrons onto the surface of a specimen, which produces images that look 3D. So very lifelike, very, very, very cool. Because of the quality the 3D SEMs produce, allows for great study of cellular structures in regards to what they actually look like. Down the bottom, the transmission electron microscopes, or the TEMs, they focus beams of electrons straight through a specimen. This allows for better abilities to study the internal structure of the subcellular components, and you can kind of see that going on in the bottom image there. I actually tried to figure out how much one of the electron microscopes cost, I came across a website for the SEMs, and for the used ones, the prices were available upon request. So I kind of translate that to mean good luck, buddy. So how do we study the individual components of cells? How do we look at the organelles and isolate what those organelles do? Well, that's where the practice of cell fractionation comes in place. Cell fractionation is used for studying those cellular structures and their functions, including the individual organelles. We can separate the organelles from one another as a result of different weights that each has. Might sound kind of strange, but each organelle weighs different masses than other organelles. So to separate these organelles, we use a centrifuge, which is really neat if you haven't used one. I've only used one for studying the hematocrit of blood, which is essentially the same as what we see going on here in the image for cells. Centrifuges spin in a circular motion at different speeds. We can adjust the speed, and also for set periods of time you can select how long you want the centrifuge to spin. So as the centrifuge spins in the circular motion, it allows for the separation of whatever substance it is that we want to examine. The heavier components will layer themselves at the bottom of the test tube, and the lighter ones will stay near the top. So we're able to actually separate them because of the different densities. I guess sort of like water and oil. If you mix those up and let them go, they'll eventually separate. It's a similar process to that. So this image shows us how centrifuges can be set at different speeds over the course of different times to obtain different results found at the bottom of the test tube for study. Looking at the first one here, it's set at a speed of 1,000 Gs, or 1,000 times the force of gravity, for 10 minutes. And we have a separation going on. This one happens to be the study of nuclei. 1,000 Gs is that's pretty insane. So that's a really good centrifuge, actually. But looking, as we get further and further down, spinning up to 150,000 Gs, and over the course of 3 hours, that's wow, that's insane. But once again, as you can see, different speeds, different amounts of time allows us to study different parts of the cell, mitochondria and chloroplasts, microsomes, ribosomes, so they are able to separate. The two main types of cells that we know of are prokaryotic and eukaryotic cells. We had spoke about those previously in this class, and they're really nothing that you should be brand new to. The image that we see on the screen is a typical bacterial cell, which are prokaryotic. Going on to the next slide, we can see some similarities between prokaryotic and eukaryotic cells. This image happens to be of a typical animal cell. These similarities will be discussed briefly, but first it should be noted that all life as we know it either does fit into one of these two categories, prokaryotic or eukaryotic. Of the three domains that we covered in Unit 1, you should recall that two of them, the domain bacteria and the domain archaea, consist entirely of prokaryotic cells. Protists, fungi, animals, and plants all consist of eukaryotic cells, so they belong to domain eukarya. Similarities that we see going on include the plasma membrane, which is right here. The plasma membrane is a selective barrier that allows oxygen, nutrients, and waste to be exchanged between the cell and the external environment of the cell. The cytosol, inside the cell, it's a semi-fluid substance that fills up the interior. Other similarities include chromosomes and ribosomes. Okay, so the general characteristics of prokaryotic cells. They don't have a nucleus. So the DNA, it's floating around in the cytosol in an unbound region called the nucleoid. There are no membrane-bound organelles either. The cytoplasm is bound by the plasma membrane. So this entire thing, it's cytoplasm inside it with the very little components that it does have. Eukaryotic cells, on the other hand, are characterized by having a nucleus, right here, which is enclosed by the nuclear envelope. Also has membrane-bound organelles. We can see a bunch of those going on around in here, the mitochondria, for example. Cytoplasm between the plasma membrane and the nucleus. So this space right here is all part of the cytoplasm. And these are generally much more large than prokaryotic cells, and they obviously have a lot more going on, so they require a little bit larger space requirements. So one of the similarities that we discussed was the plasma membrane, also known as biological membranes, also known as a lipid bilayer, also known as a phospholipid bilayer. A bunch of names for really the same thing. Looking at the plasma membranes, we can see that they're made up of those phospholipids we were talking about in Unit 2. There's a phospholipid. Phospholipids make up a double bilayer as a result of the nonpolar tails. So they're fatty, they're hydrophobic. The heads, though, those are hydrophilic, they're polar, they're water-loving. So now that we know that, why do they arrange themselves in this fashion that we see? Well, we have to recall that the body is mostly water. Even the cytoplasm, heavily water. So the exterior of the cell water, interior of the cell water, the hydrophilic heads are going to be attracted to the water, so they're kind of diverted towards it. And the hydrophobic tails are repelled by the water, but they line up great and work great next to one another. So they kind of love being with each other. They hang out, they're homies. And we have the hydrophilic heads hanging out with their big brothers, the water. And the last thing that we'll have time to talk about in this portion of the discussion is surface size to volume ratio. Yeah, it is a big deal. Alright, so we already know that cells are small. But what does the size of a cell really matter? Why is that a big deal? Functionally, small cells with a low surface area to volume ratio are more efficient at carrying out metabolic processes. This slide illustrates the concept. I do not know what the unit of measure, the 1 or 5 on the screen here represent, maybe microns. But we'll just leave them as that. I'm not really sure, so I'll just call them 1 or 5 or whatever. The surface area of the first cell is calculated by height times width, and then summing each side. So 1 times 1 times the 6 sides of the cube. This gives us the surface area of the first cube. Using the same concept for the larger cell, this cell being 5 units tall and 5 units wide, we have the surface area of one side being at 5 times 5, which equals 25. Multiply the surface area of one side by 6, each of the 6 sides, we have 6 times 25, or 150 units. Now if we break the 5 by 5 unit up into individual 1 by 1 cubes for the same space, so what makes this cube more efficient? It is broken up into these smaller units, but it's the same size as this one. Well, each side has 25 boxes, all of these 1 by 1 cubes. Since there's 6 sides, 25 times 6 equals 150, and since all 150 of those cubes have a surface area of 6, 6 times 150 gives us 750, so a much larger surface area. Now this is where the significance of cell size factors in. Because the surface areas are different for both are different, but the volumes are the same, we have a larger surface area to volume ratio. This means that the cells are in greater contact with their external environments, which allows for more nutrients, more waste products, more water, more proteins, etc. to move into or out of the cell. And it's easier. It does require less energy to be needed for this transport. I guess, provided that the process would require energy. Some processes, such as diffusion, water, that does not require energy. It's a free-moving process.